Welcome back to our session on conservation and using genetic diversity. So our next speaker is Charalambos NEOPHYTOU, who is going to talk about oaks.
So thank you for the introduction. So the title of my talk is whether adapted oaks on relict sites can contribute to future forest management in a changing climate. Actually, I'm going to present our approach in the project Aquarelle. Aquarelle means adaptation of oaks on relict sites, and it's a collaboration project between the FVA in Freiburg and AWG in Teisendorf, Bayern, and the Forest Research Institute of the Forest Enterprise of Rhineland-Palatinate. And these are our organizations that are funding this project. As an introduction, I chose a picture many of you may know. It's the spatial pattern of chloroplast DNA distribution in European white oaks. And as we can all see, we have these different colors which correspond to different lineages, different maternal lineages, and we can see that we have this lineage B, which is distributed from the western part of the Iberian Peninsula north to Denmark and England. We have this light blue lineage coming from Balkan towards west, and with red color, this Apennine lineage, as it is named. And what we can see is that they reflect post-glacial recolonization. This is due to maternal inheritance of chloroplast DNA and lack of recombination. If it's maternally inherited, it depends upon seed dispersal, and for this reason, it's not so intensively mixed. And so it reflects post-glacial recolonization pathways, but it's also kind of evidence that oaks reproduced in situ. So there is a high degree of autochthony of oak forests, at least of those forests which were analyzed in this old project, Fair Oak, which you can find also in the internet. We can also see that if we focus here in the middle, in Central Europe, we have a crossroads of different recolonization pathways. So we have a high degree of autochthony and conservation of these recolonization pathways. Let's focus a bit more on this Central European region. Here we have the region of the Upper Rhine. We have here Switzerland and Germany, the Black Forest, and France and the Vosges Mountains. And these are results from a previous project from oak stands in this area. We have here the example of Quercus robur, and we have on the left-hand side some old stands which were established more than 100 years ago. On the right-hand side, we have younger stands which were established less than 70 years ago. So mainly after the Second World War, and we can already see if we watch the pie diagrams showing the frequency of different haplotypes that there is a higher degree of mixture within stands. We can find more haplotypes within a stand if we look at younger stands. And the difference is significant. We published the results in this paper. And we interpreted this as the result of management changes. Much more was planted after the Second World War. Before we had these copies with standards management, which promoted the local gene pool. And also we had better possibilities for long-range seed transfer. So the question comes up again, where do we have really autochthonous material? This makes sites where we have really autochthonous gene pools, again, more interesting. And this is the focus of our new project, Interact. And our hypothesis is that oaks could adapt on such so-called relict sites, so sites where we assume habitat continuity. We assume that we had the same forest picture over many generations, and at that sites where oaks are autochthonous, they might have adapted over many generations. So this slide summarizes our approach in the Aquarelle project. So the first working package is, so to say, to search our research area for such putative relict populations. So we want to answer the question where such populations occur. Then we want to study habitat continuity within such populations, and we have some approaches based on insect fauna to study this. Then we investigate the gene pool of the oaks using, on the one hand, chloroplast microsatellites again. So, and we expect there, of course, to find those haplotypes that reflect the post-glacial recolonization. And we also use neutral markers to study the intraspecific structure, but also to assign individuals to species. And if the oaks have been there over many generations, they might have adapted. And to study this, we have two different approaches. We have this physiological approach, which is carried out by our partners in Rhineland-Palatinate. And we ask the question, which physiological processes are involved in local adaptation? And from the genomic point of view, we are interested in genes that carry signatures of local adaptation. So here we have a diagram which is maybe known to many people that who learned ecology, forest ecology from German sources. It shows the species that become dominant in Central Europe under specific conditions. The most common climax species is Fagus sylvatica, common beech, which dominates, which will replace ultimately other species if there is natural successions under normal pH and drought conditions. At extremely dry sites, oak species can dominate because they are too dry for Fagus sylvatica to grow. And it can become dominant there, and such sites are and putative relict populations which we are looking for grow on such sites. Also, such sites are not productive. They are too dry to get nice stems of oak and good growth. So we have no intensive management. We maybe had some grazing, but not much more, or we had some coppice management. But we believe that this management history also promoted autochthony. So these are some examples. These are two examples of such sites. Don't expect to find some nice plus trees. They are rather minus trees, I would say. We have this example here is next to the motorway. Here you have Basel in Switzerland, and it's next to the motorway. It's a calcareous cliff where the oaks grow not higher than 5 to 6 meters. This is an example of such a site. And this is another example near Staufen. It's between Freiburg and Basel. To look for such sites, we use geographic data from GIS database, which describe the site. We have site condition mapping. We also have some relict insect species, which are also documented. If we have such information, we use it, and then we visit the place and we decide whether to include it in the project or not. And these green populations are those we have already accepted and visited. Many of them were already sampled, and we have also some populations here in the north of Bavaria, which will be visited soon in order to decide whether to take them or not. As you can see, our study area begins in the Vosges Mountains in France and stretches east to the German-Austrian border and north to Oberfranken and south to the Swiss border. In many cases, we have a very different geological substrate, so we also try to have replicates within each type of geological substrate. For instance, I showed you two examples. The one is on silicate, the other one is on calcareous substrate. We have several replicates for each type of these sites. And as I said, we are interested in habitat continuity. To check for this, we carry out an analysis of beetle fauna. We cooperate with experts. I'm not the expert about this. And there are some beetle species that rely on decaying wood. And if they are present, it's a sign that there has been a habitat tradition, a habitat continuity. There has always been decaying wood on that site. Also, there are some moss and lichen species that also reflect habitat continuity, and this is another aspect which will be studied in order to investigate habitat continuity. We already carried out a pilot study using some traps, some insect traps, and we compared two sites. The one is Staufen, which I have already showed, and the other one is Falkensteig. It's to the east of Freiburg. Falkensteig is moderately dry. I forgot to translate this. And what we can see is that we have similar levels of endangered species, but in Staufen, which is warmer and drier, we found 3 so-called Urwald Reliktarten, that means primeval forest relicts. Good. This study is running. As I showed you, it's a pilot study. We decided which methods to use in the whole set of plots. The other part of the study is the investigation of origin and genetic variation, and for this we use chloroplast microsatellites and nuclear microsatellites. And we collect tissue from 48 trees per population, if this is possible. Some populations are not big enough. We decided to go down to 30, but not less. And we sample based on a systematic, based on a quadratic grid in order to systematically sample the population. Also here, our expectation is that we will find the haplotypes corresponding to the post-glacial recolonization pathway, and we expect to find a homogeneity within stands because the composition of the gene pool was shaped by post-glacial recolonization and not by seed transfer, as a stand is presumably autochthonous. And we have also here some first results for those 2 stands where the pilot study for the beetle fauna was carried out. What we can see is, on the one hand, we have the same haplotypes as the nearby stands, and in the one case we have genotype 24 trees, and all of them possess this Apennine haplotype. In the area of east of Freiburg, we found also some Iberian haplotypes, but we can see that in this area we have the same mixture also in other locations. Okay, I will try to come faster. Some words to the nuclear DNA and intraspecific structure. Of course, it is important to identify species in order to study adaptation later. So for this purpose, we have a set of nuclear microsatellites. We can see some previous results that, at least in downy oak, in Quercus pubescens, we had some intraspecific differentiation, some pronounced structure. We want to look for this also using common sets of markers. And our third hypothesis is that we had adaptation. We had high degree of adaptation. To investigate this, we have two approaches. We have the physiological approach and the genomic approach. In the genomic approach, we want to use methods that have been presented also in other talks today, mainly environmental association of variations of, from adaptive genes with environmental factors. And we also have a design to be able to carry out some outlier tests with some very contrastive sites, within gene flow distance. The colleagues in Rhineland-Palatinate study also the production of some, study also the antioxidative system and tannins with antioxidative effect. To study this, they use population pairs from contrastive sites, and they have also some preliminary results where they found that provenances from dry sites accumulate ascorbate under drought stress, which has an antioxidative effect and can help the oaks cope with drought stress, whereas provenances from moist sites have a different strategy to cope with drought stress. They accumulate two tannins, vescalagin and castalagin, in absence of drought stress, but when drought stress comes, then they can consume it to cope with it. And before this, I had our genomic approach. So what we expect is in the end to have some correlation of adaptive genes with some ecological parameters. Here I have just an example picture. And to do so, we use hybridization capture, so exome capture. So to conclude, we have here an investigation of many aspects. We first want to investigate habitat continuity by looking at the fauna and lichen and moss flora. After having studied habitat continuity, we want to see if the chloroplast DNA structure fits with an autochthonous pattern, and this leads us also to test then the hypothesis that those autochthonous oaks have also adapted on the site. And this multidisciplinary approach will help us in future answering the question whether this can contribute also to future forest management. Okay, that was it. Thank you.
We have time for one question. Then I have one myself. I wanted to ask for the genomic approaches. Are you going to start from scratch, or are you going to focus on genes, for example, that has been found under selection in oaks already?
We are planning to carry out exome capture in order to amplify, in order to investigate all annotated genes in the reference genome of Quercus robur.